Could a probiotic help prevent an autoimmune disease? A talk on the role of Lactobacillus johnsonii and 6.2 in mitigating type 1 diabetes. Type 1 diabetes is a distinct disease from the more famous type 2 diabetes, and people often confuse them. However, type 1 diabetes is an autoimmune disease. And it starts when cells in the pancreas, known as beta cells, which produce insulin and are very important for regulating blood glucose levels, are destroyed by a person's own T cells. Genetics is known to play a significant role in type 1 diabetes, but there are also significant correlations with environmental triggers such as viral infections, oxidative stress, microbiome dysbiosis, and intestinal permeability. And so this makes it a very fascinating area of study because it draws on so many kind of important areas that are complex and overlapping. Today, I want to focus on several studies about using the probiotic Lactobacillus johnsonii to mitigate type 1 diabetes. And in particular, I'll look at work from a lab at the University of Florida that has been studying this from 2010 through the present. And they've done studies involving mice, rats, human cell lines, and healthy humans. And their work is ongoing. And in particular, they have found two groups of mechanisms that are playing a role potentially in these different species, and those are a decrease in intestinal permeability as well as immune profile changes. These are in the interest of time, I can only focus on 3 studies, but there are more out there. These are the 3 studies I'll discuss today. And I'll start out with one of the earliest, which is from 2010. And this looked at biobreeding diabetes-prone rats. So this is a rat model that is very prone to diabetes. These rats were fed Lactobacillus, well, the treatment group was fed Lactobacillus johnsonii post-weaning, and that's the group up here. And this graph is showing how many did not develop diabetes. So how many remained healthy, not having diabetes. And you can see that this number is much higher with the Lactobacillus johnsonii group compared to the control group, or this group which was fed a different Lactobacillus strain. They also, I'm only going to share a few of the results, but they looked at various gene expression results and found that claudin-1, which is a tight junction protein that helps maintain integrity of the intestinal barrier through tight junctions, that those levels were higher in the L-johnsonii fed group. So here they have normalized the L-johnsonii fed group to be 1 for gene expression. And then gray is the relative expression in the healthy control, and black is the relative expression in the diabetic animals, and these levels were lower, and this was statistically significant. They also found that L-johnsonii fed rats had a higher percentage of goblet cells compared to diabetic rats. Goblet cells are cells in the intestine that produce mucus, and this intestinal mucus is another important component of the integrity of the intestinal barrier. And so these results fit with an existing body of literature on the role of intestinal integrity, gut barrier integrity in type 1 diabetes. So this diagram along the center shows cells of the intestinal lining. The top represents the lumen of the intestine, where the intestinal bacteria would be of our microbiomes. And then the bottom shows the bloodstream. And on the left-hand side of the diagram, notice that there are these gaps between the cells. And so the bacteria or different particles are able to get through these gaps. There's higher intestinal permeability here, worse integrity, and this can lead to dysfunctional immune activation here in the bloodstream. And so this is one mechanism that can be at work in type 1 diabetes. The second study I want to talk about today is from 2017, and that is the phase 1 clinical trials using this bacteria. All phase 1 clinical trials are conducted on healthy participants to test the safety and tolerance of a treatment. This was a double-blind randomized trial in which participants were given 8 weeks of daily capsules. And they were either given Lactobacillus johnsonii, or the placebo group was given capsules that just had skim milk in them. The participants received stool test, blood test, and regular questionnaires about symptoms they were experiencing. So this is the result of their weekly questionnaires. Notice on the X, it starts kind of at the baseline, then for 8 weeks of receiving capsules, and then they were monitored for an additional 4 weeks after they stopped taking the capsules. And I have put pink over 2 of the regions where there were a lot of statistically significant results. These are p-values less than 1/100th or even 1/1000th. And you can see that's particularly in the areas of abdominal pain and indigestion really being improved. And again, these were groups of healthy adults, but they saw these improvements if they were taking the johnsonii. And then this is from a daily questionnaire that asked about slightly different sets of symptoms. And here they found statistical significance of lower levels of GI distress, as well as lower cephalic symptoms. And that refers to fewer headaches and less dizziness among participants taking L-johnsonii. I did crop this graph from the paper. They looked at a number of other groups of symptoms, such as ear, nose, and throat, psychological, nausea, vomiting, but they did not see a statistical difference in those other groups of symptoms. So in conclusion, they found that those taking Lactobacillus johnsonii, that the treatment was well tolerated, that they experienced significantly less symptoms, even though these were groups of healthy adults. And then they also recorded a number of changes in immune subsets, which I don't have time to get into here, but it's fascinating to read about. And the next study gets at some of the reasons why that might be the case. So the third and final study that I'll discuss today is from 2022, and it looked at how L-johnsonii creates nanovesicles, which will be abbreviated NV in these slides. And this was conducted using human cell lines. And so they found that these nanovesicles produced by L-johnsonii reduce apoptosis rates in pancreatic beta cells. And remember, we need those pancreatic cells to create insulin, and so too much apoptosis of them, it can be a bad sign about a person's ability to produce insulin. And here they tested 2 different concentrations of nanovesicles. NV10 is the higher concentration. And they saw what happened when these cells received inflammatory cytokines. In this case, a mix of IL-1 beta, TNF alpha, and interferon gamma. And so you can see the cells receiving cytokines have higher rate of apoptosis, and those that received the cytokines together with the higher concentration of the nanovesicles had a much lower, statistically significant lower rate of apoptosis. And so that's a promising sign. And the mechanism behind this that the researchers discovered is that the nanovesicles promote AHR nuclear translocation. And so what this means, AHR is a transcription factor that influences immune cell regulation, and the nanovesicles cause this transcription factor to move to the nucleus. So the images on the left were taken at the beginning start time. Red shows where the AHR, this transcription factor, is, and it's outside the nucleus. The nucleus is in blue, and you're seeing this from 3 different angles. And then on the right, we have images of after time has passed, and you can see that the AHR has moved into the nucleus in response to the nanovesicles. And this was done in a human pancreatic beta cell line. So the researchers proposed this mechanism of action of how the L-johnsonii derived nanovesicles are impacting the human cells, and they also found that there was a shift in macrophages to a tolerogenic phenotype. So as we learned about in immunology, there are 2 types of macrophages, M1, which are pro-inflammatory, M2, which are anti-inflammatory or tolerogenic. And they found that the nanovesicles via this AHR translocation caused more expression of the M2 phenotype. And so they believe this is likely a key mechanism of what's going on. And they observed other changes related to this, such as an increase in IL-10 levels. That's an anti-inflammatory cytokine. Yeah, so in conclusion, there are a range of studies looking at Lactobacillus johnsonii and its role in type 1 diabetes. We've seen this mechanism around intestinal permeability improving, and then also how nanovesicles could be leading to immune subset changes, including promoting tolerogenic M2 macrophages and increased secretion of IL-10. And all this reduces cytokine-induced apoptosis in pancreatic cells. So the next step is phase 2 clinical trials. I know that this group is working on that. I don't know the exact status of those, but I am very, very fascinated and eager to see the results. They also highlight that this role of bacterial vesicles in cross-kingdom communication is a really emerging and exciting area of research of kind of how these vesicles from bacteria could have such an impact on humans or other species. And then I'm very curious about the kind of much longer term application or potential application of probiotics to other autoimmune diseases. These are the 3 papers I focused on today. There are many more out there if you're interested in this topic. And then I also have 3 quiz questions for you if you would like to pause your video now so that you can test your comprehension of this video. Thank you.